Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Today we will discuss the pharmaceutical technology second part in which we will now discuss the need of the pharmaceutical technology on the basis of few examples, the need for which we have actually optimized our processes of developing a new drug delivery system. Today we will actually start from that basis that how the pharmaceutical technology works in designing a new drug development, or what is the process of new drug development which is applicable in pharmaceutical technology. So first of all, whenever a new drug molecule is discovered or developed or synthesized, it has been synthesized in meansly synthetic and biosynthetic way, depending upon some docking techniques, some isolation from the plants, or some isolation from the animals, or some sources which are a combination of these bacteria, animals, plants, or a very new synthetic source. So whenever we have discovered a new chemical entity, it requires a preclinical studies. Preclinical studies involve its chemical properties, its chemistry, its structure, its molar ratios, its properties which can affect the chemical properties of the substance. Secondly, physical properties. Physical means its organoleptic properties. Secondly, its something solubilities, which are chemically physico-chemical properties, solubility studies, its behavior in the different environments, its stability when it is packed in some containers or when it is in contact with some other ingredients. These are known as physico-chemical properties of the drug. So in the preclinical study, first of all, physico-chemical properties are studied, and secondly, second studies which are known as biological studies, in which we actually establish its pharmacology, means we establish mechanism in which the drug action is produced, how it is absorbed from the body, how it will be metabolized in the body, how can it be transported from one part of the body to the target site or the disease site, and what kind of side effects it can produce, which kind of toxicity it can result when overdose will be administered, what will be LD50, what will be its ED50, means efficacious dose, 50% efficacious dose, and lethal dose, which will be 50% dose. So these are established under the preclinical studies. So under the preclinical studies, there are the studies in which we use some laboratory animals, cell lines, and other available resources to develop, to establish its biological properties. And to establish its pre-formulation studies, we use laboratory instruments and equipments so that we can find its bulk characterization, its solubility analysis, and its stability analysis. For example, we can identify the molecule, we can develop its structure, means it's a crystalline or amorphous. We should develop its solubility, in which solvent it will be soluble, whether it's soluble in the biological fluids or it is not soluble in the biological fluids, and when it will be mixed with some other excipients, some other drugs, new drug molecules, or some other ligands, whether it will remain stable or not on the basis of 5 stability parameters: physical stability, chemical stability, microbiological stability, toxicological stability, and therapeutic stability. On these basis, we will check. This is known as pre-formulation studies. Whenever a new molecule have been established as a drug molecule or have been noted as a therapeutic substance which can be used for a treatment, a mitigation, or any type of diagnostic purpose, that this drug requires to be established as a drug delivery system. So from that preclinical studies, first preclinical study, we established that this chemical molecule is now a drug substance, and now the drug substance will go for the application which is known as INDA. In FDA, it is Federal Food and Drug Administration of USA. So there is an application with the name of Investigational New Drug Application. That investigational new drug application gets a number and it is assigned to a clinical settings, a hospital, a very under strict condition environment where it will be studied for preclinical studies, plus means there will be phase 0 and phase 1 trials, clinical trials that will be conducted in the preclinical studies. And secondly, because a drug have been established, so its formulation will be developed along with us. So in the phase 0 trials, we actually use animals for the establishment of therapeutic efficacy and very specifically its toxicity. So long-term animal studies are performed to develop the toxicity or LD50 of the drug molecule. Wide pool of the animal are actually used, small animals which are used to establish its long-term toxicity studies. And then on the sideways, we actually develop its formulation. We blend it with some excipients, we coat it with some excipients, we form different formulations for oral use, for topical use, for transdermal use, for inhalation use, for parenteral use. Different formulations are prepared. For nanoparticle deliveries, microparticle deliveries are also prepared and found that it is stable in which type of the delivery system. So all the aspects are actually dealt so that an optimized formulation can be selected. For that, we develop some manufacturing and control processes. Those optimized manufacturing and control processes will be used for its formulation design. And then we design its packaging, its label conditions, its secondary packaging, and how it will be presented, and it will be stored in the environment where we are actually presenting for its therapeutic. When all the preclinical studies plus are done, then two types of trials are actually run. One are the clinical trials for INDA molecule, which is alone a therapeutic substance, drug substance without any excipients. There are phase 1, phase 2, and phase 3 trials, clinical trials, which are conducted on the drug molecule. And then on subsequent way, when a formulation has been developed, that formulation goes for the similar way trials under a controlled condition for phase 1, phase 2, and phase 3 trials. If the drug molecule initially is successful as a therapeutic substance and a non-toxic or least toxic substance when given in the therapeutic doses, then its clinical trials are established. When these three phase clinical trials have been conducted, phase 1, phase 2, and phase 3, then a new application is moved to the FDA. In Pakistan, it will be moved to DRAB for licensing and registration. So new drug application will be known as NDA. NDA form, it will be submitted under some critical protocols, means along with the formulation protocols, its packaging and label design protocols, its long-term stability studies, its phase 3 trials, phase 1, phase 2, phase 3 trials, its therapeutic efficacy, its doses, posology, and its toxicologies with mechanism of action, with the duration of action and duration of the therapy. The DRAP will actually review it, or it will check it that whether it having all the parameters, then a plant visit will be conducted and approval of the plant will be given where this formulation will be prepared. So an inspection will be made for plant approval, and then if the plant is approved, the facilities are approved at which this formulation will be prepared, then FDA will give a specific number on the NDA form, which is known as registration number of the product, and plant licensing number have been issued to the plant that will be responsible for the production of this registered numbered product under the license number which is given to the plant. So this registration license number and registration number will be now fixed on the packaging design along with the price. Price will be nominated on the packaging design, and it will be marketed in a specific community, in a wider community where phase 4 clinical trials will be performed. Phase 4 clinical trials actually are known as post-marketing surveillance, in which a wider population will be used to establish clinical pharmacology, clinical toxicology, and any other indication that will appear other than demonstrated in the INDA form. That will be documented also. Any adverse drug reaction which are reported, which were not indicated in the initial process INDA form, will be noted down. And similarly, any type of deformity or defects that appear in the formulation or long-term storage and long-term use will be reported as product defect reporting. And then product line extensions are also reported. After post-marketing surveillance, the new drug is established. So this new drug development process, if it is performed for any new drug entity, then that drug will be an optimized formulation, and optimized formulation is a main aim of our pharmaceutical technology.